All right, folks. So we just finished the first step of cellular respiration where glucose got converted to pyruvate. We got two NADs going over to that last and final step, and we'll see what happens to them later. And we also produced two ultimate ATPs. But that pyruvate now is going to go into the next step. And notice it's going into the mitochondria. Think of it only able to go into the mitochondria, though, if oxygen's present. And we'll talk about oxygen and its very important role with pulling these electrons through, pulling all this through. So now we're going to focus on this next step, pyruvate oxidation, or what I call the conversion of pyruvic acid, or conversion of pyruvate. Okay. And here I have a picture. And look, isn't this so nice? It goes so fast. All right. Here was the pyruvate. Notice it's a 3-carbon compound. This is going to come in very important here. 1, 2, 3 carbons. That enters through a transport protein, the membrane of the mitochondria, into the matrix of the mitochondria. When that happens, it loses a carbon dioxide. If you lose a carbon dioxide, then you're down to 2 carbons. See, the 2 carbons. And now this is no longer pyruvate, but it's called acetyl coenzyme A. Now it's coenzyme A because not only are you losing carbon dioxide, but you're also attaching a coenzyme to it. Remember, coenzymes were little fixers for enzymes. They kind of temporarily attach so that enzymes can work better. So it temporarily attaches to this acetyl group. This is just the acetyl here. We call it acetyl coenzyme A because it has the coenzyme temporarily attached to it. With all that happening, there goes a NAD. That's why I kind of don't like this diagram sometimes. I wish they had one more like white arrow. I'm going to draw it kind of going like this over to here. Okay, I'm going to draw kind of an arrow in here. Boop, 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 boop. And I wish that was white. I should get my whiteout tape. Ooh, that would look really good, I think. Okay, so if I had my whiteout tape and I was, come on, whiteout tape, work for me. And I had an arrow also leading. Oh yeah, I'm messing this all up and you guys are witnessing it firsthand. Okay, come on, tape. All right, it's not going to work for me. Try over here. Oh, this tape. Sometimes it's like a blessing and sometimes it's just like, what are you doing, tape? Might be at the end of it too. Who knows? Let's see. Try this over here. Is it working? I don't think my whiteout tape is working anymore. Alrighty. Oh, wait, I got it working, you guys. Jeez, here you are watching me like struggle with all my little like office supplies. Oh, God, I can't get this to work at all. All right, bad idea, you guys. I'm not going to sit here and struggle with that. But imagine that there is another arrow, okay? Because they have 2 NADs coming from this. Well, you know what? There's also, hold on here, there are also 2 NADHs coming from here. Wait a second. I see only 1 coming from here. Well, you know what? Let me get this centered here, okay? Why am I saying there's 2 coming from this? Okay, there's only 1 here. Okay, you know why I'm saying that? Because 2 times we had 2 pyruvates formed. Let me bring back glycolysis and show you. 2 pyruvates were formed at the end of glycolysis. So each one is going to have to go through this. One through it and the other one through it. So anything we do times 2. Okay, this whole step is going to go twice because there were 2 pyruvates coming in. All right. So one more time. The pyruvate that has 3 carbons loses carbon dioxide. Okay, so it takes it down to 2 carbons, attaches a coenzyme to it to make things go easier. And then there go those NADs again. Okay, 1 per each pyruvate, but everything happens twice. Okay, so if everything happened twice, then how much carbon dioxide did we get out of here? Yes, we got 1 for the first pyruvate, 1 for the second. So we also got 2 times the carbon dioxide out of here. And that's that second step. Okay, so go back to my messy like thing here. Our first step, we got 2 NADs coming out, going over there, and we got 2 ATPs. We ultimately changed glucose to pyruvate. Second step, we changed pyruvate to acetyl coenzyme A. We got 2 NADs out and we got some carbon dioxide, but no ATPs. Okay. And I could say, like, oh, well, we got a little bit of carbon dioxide out of that, 2 CO2s. Okay. The school's playing some music here. Just ignore the music in the background. Okay. 2 CO2s out of here. That's okay. But those gnats are really important. I'm kind of sad that they didn't have that third arrow going in there. Okay. All right. So now let's go on to the next thing. So we're going to take that acetyl coenzyme A and we're going to head into the Krebs cycle with it. Okay. The Krebs cycle is going to look quite confusing. There's going to be a lot of molecules involved in that, but not as confusing as this yet. So bear with me. We are getting to that final thing. So far we've only gotten 2 ATPs. We've gotten some carbon dioxide out. We have a bunch of NADs going over there. Let's take a look at the Krebs cycle. So let me take away this pyruvate and we'll put up the Krebs cycle. Wait till you see this. Bum, ba, da, bum. And by the way, you're going to get these diagrams. Okay. This looks super confusing. I know it does. I know. But it's not going to be, because number one, you do not have to sit here and memorize all these words. Let me just fix this a little bit. It's driving me nuts. All right. Here comes the acetyl coenzyme A. Let's start over here. Remember, it has 2 carbons. That's one thing I definitely want you to pay attention to, the number of carbons. Here is this substance called oxaloacetate. Oxaloacetic acid, we used to call it. Now they call it oxaloacetate. Look at how many carbons. 1, 2, 3, 4. Okay. 2 plus 4 makes 6 carbons. Where do you see the 6? 1, 2, 3, 4, 5, 6. This is citrate, or better known as good old citric acid. Same thing. Okay. And like I said, they used to call all these 8s ic acids. They called this citric acid, isocitric acid, ketoglutaric acid, succinic acid, succinyl-CoA, succinic acid, fumaric acid, malic acid, oxaloacetic acid. And now they call them all 8s. Okay. So now we have 6 carbons. Okay. This came from the conversion of acetyl coenzyme A, or conversion of pyruvate, right? Entered in, added to this. So 4 plus a 2 makes a 6. And this is why they call it the citric acid cycle, because it's the first component in this cycle. Okay. Citric acid will convert to isocitrate. It's an isomer of citric acid. That's all it is. It's just rearranged. Okay. And you can see how it's rearranged if you look closely, but you know what? I don't care for you to know that. But look at what happens. It loses carbon dioxide right here. Carbon dioxide comes out. Okay. If it loses a carbon dioxide, then it's going to go down to only 5 carbons. But look at what else goes through. There's those NADs again, stealing electrons and taking them over to the electron transport chain, or oxidative phosphorylation. Now we have ketoglutaric acid, ketoglutarate. Loses another carbon dioxide, and there go those NADs again. NADs here, NADs there. And we're down to a 4-carbon compound called succinyl. Well, look at what happens here. A little ATP is formed. Don't worry about the GTP. Okay, ATP is adenosine triphosphate. GTP is guanine triphosphate. Not a big deal. Don't worry about it. It's very similar to ATP. We're not going to stress about this. But we get ATP out of this. Yay! Yay! I almost want to start all around. We get ATP out of this. And now we're down to succinyl acid. Succinate. Excuse me. Then look at what happens. We don't lose any carbon dioxide. There's 4 carbons. Didn't lose any carbon dioxide over there. 4 carbons. But there goes a fad. A fad is like a NAD. It's going to carry electrons over to oxidative phosphorylation. So we had 4, but we didn't lose any carbon dioxide, so we still have 4. So we have fumaric acid. Yes, a little bit of water goes in here, comes out here, or goes in here. Don't worry about the waters just yet either, okay? But now we're down to malic acid. Look, there go more NADs, okay? But notice we didn't lose any carbon dioxide, so we're still 4 carbons, 4 carbons, 4 carbons, but now we're back to that oxaloacetic acid, which will pick up another acetyl and go around again. And you know it's going to go around 2 times. because remember we had 2 pyruvates that changed to 2 acetyl coenzyme A's. So we got to go around twice. So everything that's happening here, if we have 1, 2, 3 NADs, it's times 2. So we actually have 6 NADs. And if we have 1 fad, it's times 2 because we have 2 fads. And if we produce 1 ATP, it's times 2. So we actually got 2 ATPs. So let's look at our totals going on here. Okay, our main cycles, right? And 2 NADs came from glycolysis, 2 NADs came from that. Well, look, 2 NADs here. Yeah, we have 6. I'm going to put a 6. I know that's tiny and you can hardly see that, but there's a 6 right next to the NADs, and 2 next to the fads are going over to oxidative phosphorylation. And how many ATPs? We got 2 ATPs out. And we also got some carbon dioxide. Let's see how much carbon dioxide we had out. Where's that diagram? It's right below here. Okay, we had a carbon dioxide come out here and a carbon dioxide come out here. That's 2 times 2 apiece. So we had 4 CO2s come out. Remember how we said in the main reaction we were going to produce 6 carbon dioxides? There they are, 4 here, 2 here. The pyruvate oxidation and citric acid cycle are where you lose carbon dioxide to breathe out. Carbon dioxide. That's where it comes from, okay? And also from that acid blood. Remember how we were always dropping acid? But a little bit comes out of this, okay? This is not all the carbon dioxide that's produced from your body. Remember we learned in the beginning of the year that most of the carbon dioxide produced is really to like neutralize your blood, okay? Yeah, some of you might remember that. Okay, you know what's driving me nuts is that I didn't finish that white arrow there. Yeah, I know, I'm bringing it up again. All right, so what have we got here? First step gave us 2 ATPs, 2 NADs heading over there. Second step, no ATPs, but we released carbon dioxide and it gave us 2 NADs heading over there. Third step gave us 6 NADs, 2 fads heading over to there, released some carbon dioxide and gave us 2 ATPs. Ooh, Krebs cycle. I totally think they're winning right now, okay? And now we're going to go to the big payoff. That's the oxidative phosphorylation. And that's going to be our next video. Okay, but just want to point out here that this is not going to happen twice because if you remember, we're done with the 2 pyruvates entered in here. We had 2 acetyl coenzyme A's. This goes around twice, churns out these, but it's not going to pass on any substance because remember, it ends up being that oxaloacetate that gets just recycled in. So nothing's moving on except for the NADs and the fads. Okay, so this is not going to happen twice. So when we go through oxidative phosphorylation, we're actually going to follow what one NAD will do and how ATPs will be produced. And then we'll kind of count up the NADs at the end to see like how many ATPs do we really get out of this. Okay, so we got 2 so far, 2 so far. It's going to be a big payoff, you guys. Okay, and that's the next video.